Hello friends, are you preparing for your Edexcel GCSE biology exam? We got you covered. We are making a series of short, circa 20-minute videos to cover the 9 main topics for biology and help you get your top grade sorted. If you start your revision using these videos and are able to make the time to watch 2 each week, you will cover all the biology content in 5 weeks. Please do like and subscribe to our channel if you found this video useful, and do share with your classmates. We start with diving into a crucial topic for your Edexcel GCSE biology exam, cell biology. In this video, we will cover the basics of cell biology, mitosis and meiosis, microscopes and stem cells. Cell biology. This is a fundamental area that covers the very basics of life, how cells function, and how they divide. And there is always a question on this topic in the exam. By the end of this video, you'll be fully prepared to tackle any cell biology question with confidence. So let's get started. Cell structures and functions. At the very core of cell biology, we need to understand what makes up a cell. Every living organism is made up of cells, and all cells come in 2 types, prokaryotic and eukaryotic. Prokaryotic cells, like bacteria, don't have a nucleus, while eukaryotic cells, like those in plants and animals, do have a nucleus. For a top grade, you will need to explain in detail the differences between these 2 types of cells. Prokaryotic cells and eukaryotic cells differ in several key structural aspects, reflecting their evolutionary divergence and distinct roles in the biological world. Prokaryotic cells are typically simpler and smaller than eukaryotic cells. They lack a true nucleus. Instead, their genetic material is found in a single circular DNA molecule located in the nucleoid region. Prokaryotes do not have membrane-bound organelles, meaning structures like the mitochondria, endoplasmic reticulum, and Golgi apparatus are absent. The cell structure of prokaryotes often includes a cell wall composed of peptidoglycan in bacteria, which provides shape and protection. Additionally, many prokaryotic cells have flagella for movement and pili for attachment to surfaces or to other cells. An example of a prokaryotic cell is a bacterium, such as Escherichia coli, E. coli. Eukaryotic cells, on the other hand, are generally larger and more complex. They have a true nucleus enclosed by a nuclear membrane, which houses their linear chromosomal DNA. Eukaryotic cells contain a variety of membrane-bound organelles that perform specialized functions. For example, mitochondria generate energy through aerobic respiration, while the endoplasmic reticulum and Golgi apparatus are involved in protein and lipid synthesis and processing. Plant cells, a type of eukaryotic cell, have additional features such as chloroplasts for photosynthesis and a cell wall made of cellulose. Examples of eukaryotic cells include animal cells, such as human liver cells, and plant cells, such as leaf cells. In summary, prokaryotic cells are structurally simpler, lacking a nucleus and membrane-bound organelles, while eukaryotic cells are more complex with compartmentalized functions due to their organelles. The structural differences are reflected in their size, complexity, and roles in different organisms, with examples being bacteria for prokaryotes and animal or plant cells for eukaryotes. Let's look at the structures found in cells in more detail. Mitochondria. The mitochondria are known as the powerhouses of the cell. Their primary function is to produce energy through a process called aerobic respiration. This process converts glucose and oxygen into adenosine triphosphate, ATP, which is the energy currency used by cells to carry out various metabolic functions. Mitochondria have a double membrane structure, with the inner membrane folded into structures called cristae. These folds increase the surface area for the electron transport chain, which is crucial for ATP production. Cells with high energy demands, such as muscle cells, contain a large number of mitochondria, because they require significant amounts of ATP to function effectively. For example, skeletal muscle cells contract and relax continuously during physical activity, requiring a constant and abundant supply of energy. Cardiac muscle cells also have high energy requirements to maintain the rhythmic contractions of the heart. The large number of mitochondria in these cell types ensures that the cells can produce sufficient ATP to meet their energy demands, enabling sustained muscle contraction and optimal function. Without enough mitochondria, these cells would quickly deplete their energy stores, leading to muscle fatigue or impaired function. The cell wall and cell membrane. The cell membrane, not to be confused with the cell wall, is a barrier that controls what enters and exits the cell. The cell wall is present in plant cells, but absent in animal cells, leading to significant differences in structure and function between these cell types. The plant cell wall. The cell wall in plant cells is a rigid outer layer that surrounds the cell membrane. It is primarily made of cellulose, a strong carbohydrate polymer that provides structural support, shape, and protection to the cell. The plant cell wall usually consists of 3 layers: the primary cell wall, the secondary cell wall, sometimes not present, and the middle lamella that helps bind adjacent plant cells together. Its functions include maintaining cell shape, resisting mechanical stress, controlling cell growth, and regulating the movement of water and nutrients. The rigidity of the cell wall allows plants to stand upright and grow towards light. Animal cells. Unlike plant cells, animal cells do not have a cell wall. Instead, they have a flexible cell membrane, also called the plasma membrane, that provides a protective boundary and controls the movement of substances in and out of the cell. The absence of a rigid cell wall makes animal cells more flexible and capable of taking on a variety of shapes. This flexibility allows for specialized functions, such as muscle contraction and the formation of different tissue structures. Key differences to note. Presence. The cell wall is present in plant cells but absent in animal cells. Composition. The plant cell wall is mainly composed of cellulose, while animal cells have only a flexible cell membrane. Function. The plant cell wall provides structural support, protection, and rigidity, whereas animal cells rely on their flexible cell membrane for protection and communication with the environment. In summary, the presence of a cell wall in plant cells contributes to their structural rigidity and ability to withstand turgor pressure, while the absence of a cell wall in animal cells allows for greater flexibility and dynamic functions. All these organelles work together to keep the cell functioning properly, whether it's a single-celled organism, a plant cell, or a cell in a human body. You will need to be able to identify these structures in each cell type and label them, so make sure you are familiar with the structures found in these types of cells. Now let's move on to how cells divide. Cell division, mitosis and meiosis. Understanding cell division. Cells divide through 2 distinct processes, mitosis and meiosis. First, let's talk about mitosis, which is responsible for growth and repair. Mitosis starts with a single parent cell. The chromosomes inside the nucleus duplicate, and the cell splits into 2 identical daughter cells. Each daughter cell has the same number of chromosomes as the parent cell. This is essential for growth and tissue repair. There are 6 distinct phases of mitosis. Understanding and explaining the stages of mitosis is a key component of the Edexcel GCSE biology curriculum and is commonly assessed in exams. Let's examine each one. Interphase. The cell spends most of its life in this phase. The DNA and chromosomes copies itself ready for mitosis. Prophase. The DNA and chromosomes and their copies condenses to become more visible. The membrane around the nucleus disappears. Metaphase. Chromosomes and their copies line up in the middle of the cell. Anaphase. Chromosomes and their copies are pulled to different ends of the cell. Telophase. New membranes form around the chromosomes at each end of the cell. Cytokinesis. The cell membrane pinches in and eventually divides into 2 daughter cells. Here's a quick mnemonic to help you remember the stages of mitosis in order. I prefer meatloaf at the cafeteria. This mnemonic breaks down each stage of mitosis and helps you recall the sequence quickly and accurately. Common exam questions may require you to do the following. Identify and describe stages. Recognize and explain each phase of mitosis and detail the key events in each phase. Sequence stages. You may be asked to arrange the stages of mitosis in their correct order. Interpret diagrams. Analyze diagrams or micrographs depicting cells in various mitotic stages and identify the specific phase shown. Explain the significance of mitosis. For example, discuss the importance of mitosis in growth, repair, and asexual reproduction. So, do make sure you are familiar with these processes and their significance. Now, meiosis is a bit different. It's used to produce gametes for sexual reproduction. It uses the process of meiosis, which creates gametes, sperm and eggs, ova in animals, and pollen and ova in plants. Unlike mitosis, meiosis results in 4 non-identical cells, each with half the number of chromosomes. The process of meiosis happens in the male and female reproductive organs. As a cell divides to form gametes, copies of the genetic information are made. The cell divides twice to form 4 gametes, each with a single set of chromosomes. Gametes are haploid and are genetically different from each other. Haploid describes a cell that contains a single set of chromosomes and can also refer to the number of chromosomes in egg or sperm cells or gametes. In humans, gametes are haploid cells that contain 23 chromosomes, each of which becomes one of a chromosome pair that exists in diploid cells. This leads to genetic variation, which is crucial for sexual reproduction. Here is a typical exam question about meiosis from a past paper and its top grade answer. Explain how meiosis leads to genetic variation in gametes. Meiosis contributes to genetic variation in gametes through 2 key mechanisms, which lead to the production of 4 genetically different haploid cells, each containing half the number of chromosomes as the original cell. Crossing over. During meiosis, homologous chromosomes can exchange segments of DNA, resulting in a process called crossing over. This creates new combinations of alleles on each chromosome, increasing genetic diversity in the resulting gametes. Independent assortment. In meiosis, homologous chromosome pairs align randomly. This random orientation means the distribution of chromosomes to the gametes varies, leading to different combinations of maternal and paternal chromosomes. These mechanisms result in 4 genetically unique haploid gametes, contributing to the genetic variation observed in sexually reproducing organisms. These mechanisms ensure that each gamete produced is genetically unique, contributing to the genetic variation observed in sexually reproducing populations. Although detailed knowledge of the stages of meiosis is not required for your exam, understanding its role is important for appreciating genetic diversity. Microscopy and magnification. To study cells in detail, we use microscopes. You've probably used a light microscope in school, but scientists often use electron microscopes for more detailed observations. These microscopes have much higher magnification and resolution, allowing us to see things like the intricate structures inside mitochondria. Let's talk about magnification. Here's the formula for magnification. Magnification equals image size, divided by actual size of the object. For example, if you have an image that's 10 millimeters long, and the actual cell is 0.01 millimeters, the magnification would be 10 divided by 0.01, which equals 1,000 times. Make sure you're comfortable with this calculation, as it does tend to come up in exams. Stem cells and their applications. Let's now look at some very special cells. Stem cells. Stem cells are undifferentiated cells, meaning they can become any type of cell in the body. They're incredibly important in medicine because they have the potential to repair damaged tissues or even grow organs. There are 2 types of stem cells, embryonic and adult stem cells. Embryonic stem cells are versatile. They can turn into any cell type. Adult stem cells are more limited but can still regenerate tissues, like bone marrow producing blood cells. However, The use of embryonic stem cells is controversial due to ethical concerns, as it involves the destruction of embryos. Writing a top-grade answer. Now let's talk about how to make sure you can write a top-grade answer in your biology exam. The key to getting the highest marks is to demonstrate detailed knowledge and critical thinking. Your answers should use precise terminology, include detailed descriptions or explanations, especially of processes like mitosis or stem cell use. Provide examples where relevant. Address any ethical, social, or environmental implications if the question asks for it. And be structured clearly. Start with a definition or introduction, followed by the main body of your explanation, and finish with a conclusion or evaluation. If you're a student aiming for top grades in biology, you will need to practice answering exam questions. Be sure to share, like, subscribe, and hit the notification bell for more exam tips and subject breakdowns. Following on from this, we will have a video of just past paper questions on these topics, which will help you to revise, so do look out for it. Please do give your thoughts and comments below, as we value your feedback on our content. Thank you for joining us. Like, subscribe, and share. And again, look out for our next video.